cheio, mano. Tá até cheio. Essa luz na cara é insuportável, né?
É, você fica meio...
A gente fica aqui em cima ou desce, hein? Fica aqui em cima, né?
Fica aqui mesmo.
É, a gente espera.
Ah, tem que esperar?
Não, acho que... Are we good? Yeah, we're good to go.
So, good morning, ladies and gentlemen. Thank you very much for joining us today. Targeting inflammation in infectious diseases is the title of this symposium, which the Brazilian Society of Pharmacology and Experimental Therapeutics, SBEFT, has organized. I also would like to thank the International Organizing Committee for accepting our proposal and for the opportunity to showcase in the WCP 2023 advanced research on mechanisms by which inflammation could be targeted to prevent tissue injury and preserve homeostasis in infectious diseases. Different microorganisms can subvert the human immune system, causing various infectious diseases. Bacteria and viruses are the most prevalent, but protozoa and parasitic infections are also health problems of great concern. The immune system represents the host defense system. If the immune system barrier is disrupted, inflammation is the result and the body's first reaction to infection. Thus, inflammation protects the host from infections by eliminating pathogens and promoting tissue repair and recovery. However, shifts in the inflammatory response from short to long-lived can lead to significant pathological change and cause disease. I am Marco Aurelio Martins, researcher of the Laboratory of Inflammation of the Oswaldo Cruz Foundation, Brazil, and current president of the Brazilian Society of Pharmacology and Experimental Therapeutics. I will co-chair this session with Mauro M Teixeira, a professor of immunology at the Department of Biochemistry and Immunology of the Federal University of Minas Gerais, Brazil. Mauro, the floor is yours.
I'll say good morning, everyone. It's a pleasure to be here, and thank you for coming. It's nice to talk about infection and inflammation, and especially in the post-COVID area, where all of a sudden people became aware infections are relevant again. So we'll have a slight change in order. One of our speakers actually missed his connections to Glasgow, so and if you're coming from Brazil, if you miss your connection, you lose a day. So there's no other way you can do that. So our first speaker will be Patricia Bozza. Patricia is from Oswaldo Cruz Foundation. She's a brilliant pharmacologist, currently vice president of the Brazilian Academy of Science. And she will be talking to us about her beautiful work on thromboinflammation and COVID-19. Thank you, Patricia.
So thank you very much, Mauro, for the kind introduction. It's really a great pleasure to be here and to be able to share with you some of our work on the roles of thromboinflammation in COVID-19. And I'd like to especially thank the organizers for this opportunity and the Brazilian Society of Pharmacology, and I'd like to thank Marco Aurelio Martins for this kind invitation to be here. So my talk today will be focused on both the acute part of COVID-19 and how thromboinflammation participates in mechanisms involved in severity of disease. And in the second part of my talk, I will focus on on the post-COVID aspects of the disease and how thromboinflammation are activated and may participate in some of the mechanisms involved in the post-COVID aspects and pathogenesis. So as we enter in the 4th year of COVID, COVID-19 still presents quite a significant challenge for patients, families, and health systems globally, and that is the information for this past week. It's been, there have been more than 700 million confirmed cases of COVID-19, including over 6 million deaths. And in Brazil, we have quite a high burden of disease with more than 37 million cases confirmed and more than 700,000 deaths. Before vaccination, most COVID cases would have mild to moderate symptoms. However, approximately 15% of the cases would evolve to more severe cases of disease and leading to ARDS, septic shock, and eventually quite a number of deaths. Vaccination has completely changed this situation, and that's why we are here back in a presential meeting today. Vaccination has dramatically reduced the number of severe cases and mortalities for COVID, and also, as we are going to see more related to the second part of the talk, vaccination is also protective for the post-COVID syndrome. So even though we are now in a much better situation that we can gather here together without masks, concerns persist regarding both viral variants as well as the long-term consequences of COVID. So our lab from the beginning were interested in understanding the immune inflammatory mechanisms associated with disease severity, also the mechanisms of host-virus interaction and the metabolic aspects of these interactions, development and repurposing of antivirals, and more recently, we are now involved in addressing long-term consequences of COVID. Today, I'd like to focus my talk on the role of thromboinflammation, both as mechanisms involved in the severity of disease and potentially also involved in the long-term consequences of disease. And I will discuss with you some of published results and some results that are still ongoing. So since the beginning of COVID-19, it became quite clear that COVID-19 exhibit markers of hypercoagulability and thrombosis. So markers of coagulation including increased levels of D-dimer has been associated with severity and mortality, and that results came just from the beginning of the pandemics, and the observations that there were quite a high prevalence of venous and arterial thromboembolic events, and as we can appreciate, and I'm not sure if I can get the pointer here. Well, we can appreciate the microvascular thrombi in autopsies of fatal cases of COVID. So indeed, quite recently, as part of a project, a sub-project of ISAREC. ISAREC is a large group of international group to study severe acute respiratory and emerging infection. We had the opportunity to revise 495,000 cases of COVID worldwide. That is data from 55 countries. And we addressed the question, so is coagulopathy indeed important for severity and mortality of COVID? And most importantly, as well pointed out in the keynote speakers yesterday, there are a lack of data for low and middle income country to assess those information. So part of our question was to compare the data between high-income countries and low-income countries. And indeed, our data indicated that although high-income countries exhibit higher frequency of coagulopathy and complications, low and middle income countries exhibit a higher mortality when those coagulopathy complications are present. So as we can appreciate in this figure, indeed, coagulopathy complications is an independent risk for mortality of COVID-19. And as I said, COVID-19 was not equal around the globe. So low and middle income countries and poverty and inequality played also quite a big role on COVID, and that can be appreciated here in this figure, that the mortality associated with coagulopathy is much higher in countries or in the population from low and middle income countries. So we've been interested to understand the mechanisms that are involved in this thromboinflammation that occurs in COVID, and how are those hypercoagulability controlled. And now we have quite a good sense that these mechanisms are multifactorial. They involved both activation of different cells and systems and molecules. And what I would like to point to you today, and that's our discussion today, that platelets do play a very important role in the mechanisms that orchestrate the response in terms of thromboinflammation, both by the way that they can be activated and secrete different mediator, but also by the aspects that they can communicate and modify the response of other leukocytes and endothelial cells, as well as drive the activation of the extrinsic coagulation pathway. So what I would like to point to today is that platelets are indeed activated and inhibit hyperreactivity during COVID. Platelet leukocyte aggregates are increased in COVID-19, and it's associated with disease severity, and it indeed has roles in hypercoagulability and also in the amplification of the inflammation. So platelets are indeed key cells in hemostasia and vascular stability and vascular repair. But over the past years, it became quite important that platelets are indeed major effector cells in inflammation and immunity. So platelets can be rapidly activated and upon activation, platelets can release a number of cytokines, chemokines, growth factors that are stored in their granules, as well as to express different molecules. And I'm going to use here as a marker of activation the expression of P-selectin upon platelet activation, as well as platelet can produce newly formed mediators, including lipid mediators, as well as exhibit signal-dependent splicing and translation of RNA transcripts, including for cytokines and tissue factor. But also, and quite important for our discussion today, platelets also exhibit their effect by their interaction with different leukocytes, mostly monocytes and neutrophils. And in that way, platelets can modify the phenotype of those cells, so can modify the phenotype of monocytes, as well as in the case of neutrophils, it's been quite well demonstrated that platelets can induce netosis by the neutrophils. Our hypothesis is that platelets are circulating pathogen sensors that can rapidly respond and be effectors of inflammation in systemic infectious diseases. That's been demonstrated in different infections by different groups, including in malaria, dengue, sepsis, and we've asked what were the roles of platelets in COVID-19, especially to their roles in thrombosis, as well as in the amplification of the inflammatory response. So to summarize quite a high volume of data, we and others demonstrated that platelets are indeed activated during COVID. That can occur directly by the interaction with the virus, as well as indirectly by the inflammatory milieu that induces platelet-derived inflammatory mediator secretion, and that can occur importantly in the lung. Also, platelets exhibit hyperreactivity, so platelets from COVID patients respond to very low concentrations of other stimuli that in normal subjects that would not induce platelet activation, and also that platelet gene expression and proteome is dramatically altered during infection, and platelets also interact with leukocytes, and here I'm showing the data for platelet interaction with different leukocytes, and we can appreciate here an increased in platelet monocyte interaction during COVID-19, especially in the severe forms of disease that we can see the monocytes here that are decorated with platelets interacting closely with the monocyte. Also, platelet can, in COVID-19, interact with neutrophils, and as we are going to see, those interactions with both the monocyte and neutrophils would have roles in altering the phenotype of those cells and may contribute to pathogenesis of disease. So we first asked, would platelets have roles in inducing tissue factor expression. So tissue factor is a key molecule to activate the extrinsic pathway of coagulation. And also, tissue factor has many roles in inflammation. It can be driven by inflammatory response, but can also activate and perpetuate the inflammatory response. So we went on to see if platelets could modulate the expression of tissue factor by monocytes, and here is one of the examples of these findings, that here we can have platelets that are marked with CD41, and human monocytes from COVID or healthy subjects were labeled with CD14. So in here, to simplify the data, I'm showing monocytes from severe form of COVID-19, and we can see that the monocytes from those patients, the ones that have platelets decorating those monocytes, are the ones that express the highest levels of tissue factor. So to see if indeed platelets were the responsible for this modulation, we did an ex vivo assay where platelets from severe COVID-19 patients or platelets from health subjects were put to interact with monocytes from healthy subjects. And as we can appreciate here, platelets from COVID-19 patients triggered increased tissue factor expression in monocytes from healthy donors, an effect that could not be seen when heterologous platelets from healthy individuals were used. We went on to identify what were the mechanisms involved in this interaction, and we demonstrated that this interaction involves both P-selectin and integrin alpha-2 beta-3 signaling to trigger the monocyte tissue factor increased expression. In addition, platelets also release platelet-derived extracellular vesicles enriched in tissue factor, and that can also be a way to activate the coagulation system. And as we can appreciate here, severe COVID-19 patients exhibit increased levels of those tissue factor or procoagulant vesicles in the circulation, and mostly those tissue factor exhibiting extracellular vesicles, they associated with a poor outcome that are increased in patients that have not survived COVID-19 infection. Unfortunately, Fernando Cunha could not attend the meeting. As Mauro said, he missed the plane. But another very important part of COVID-19 pathogenesis, and the group led by Fernando Cunha was the one to first demonstrate that NETosis do play a very important role in the pathogenesis of disease. And here is a study by Fernando that we can see that NETs are increased in the tracheal fluid from COVID-19 severe patients, and also that NETs are formed in the lung samples from patients that from fatal cases of COVID-19. And indeed, different groups, including the group led by Rob Campbell, demonstrated that indeed to have net formation, platelets do play a key role. So as we see for the monocytes, platelet also interact with neutrophils. We can observe here a thrombi in the lung of a patient of a fatal case of COVID, that this thrombi, they exhibit neutrophil platelets and signs of citrullinate histone, demonstrating the presence of platelet-neutrophil interaction and netosis under this situation, and that, as we've shown in those mechanisms of platelet from COVID inducing activated of the monocytes, similar results can be shown here by the group of Nicolai and collaborators, that platelet from COVID-19 interact with neutrophils, and that is essential for the increased net formation in this case. And indeed, platelet activation and tissue factor expression are predictors of severity and mortality in COVID, and we can appreciate here data when patients that have those different parameters analyzed within the 24 hours of hospitalization, they are predictors of mortality in the case of P-selectin, soluble P-selectin, and also of tissue factor expression. So the mechanisms involved, and what I've shown to you, is that increased platelet activation and platelet leukocyte aggregation are observed in severe forms of COVID. Platelet activation licensed the monocyte for tissue factor expression and for increased extracellular vesicle, procoagulant extracellular vesicle with tissue factor expression, and also that platelets trigger nets and also tissue factor enriched nets in COVID. So we next asked if would those interactions of platelets and monocytes also contribute to the increased inflammatory response, or to the dysregulated inflammatory response that we see in the severe forms of disease? And the response to this question is yes. So using that same model of using platelets from severe forms of COVID disease and heterologous interactions with either monocytes or platelets from health subjects, we can appreciate here that platelets from COVID-19 patients, when interact with monocytes from healthy donor, that leads to an increased production of cytokines, including TNF, and also cytokines that are involved or that are derived upon inflammasome activation as IL-1 beta. Also, interestingly, this interaction is reciprocally, so monocytes that are activated in COVID can also activate and recruit more platelets and increase the response of platelets from health donors. So, and that is shown that the interaction of monocytes from COVID patients can lead to an increased secretion of PDGF by platelets from a healthy individual. We went on to sort out what would be the mechanisms involved in those amplified inflammatory mediator secretion, and our data point out to a very important role of that involves the interaction of the platelet with the monocyte, again, through both P-selectin and alpha-2b beta-3 integrin, but also tissue factor has a quite important mechanisms acting at part 2 on regulating this amplification of inflammatory secretion, as we can observe here by the inhibition using an antibody that blocks the signaling domain of tissue factor that leads to an inhibition of the pro-inflammatory secretion by the monocyte. In addition, we can block that with PAR antagonists. So, as I've pointed to you, those interactions, they are very important and multimediated to both, and platelets play a key role in this amplification of the inflammatory response, both by their activation as well as by the interaction of different cells. And to summarize this part, the interaction of platelets with monocytes and neutrophils, they amplify the thromboinflammatory response in COVID-19, and that is very important to contribute to the state of hypercoagulability and thus in severe forms of disease. So in the last part of the talk, I would like to discuss with you some of more recent data on the long-term consequences of COVID-19. So post-acute COVID-19 syndrome, it refers to quite a diverse group of signs and symptoms. More than 200 symptoms has been described that some people experience after having COVID-19. Some symptoms persist after the COVID-19. Some symptoms appear after the COVID-19 onset of disease. The most important group of symptoms are the ones from the nervous system, as we've heard early in this morning. So memory loss, stroke, and foggy thoughts are quite important symptoms related to the long-term consequences of COVID, together with depression, anxiety, and mental health disorders. In addition, blood disorders and especially thromboinflammatory mechanisms and thrombosis, thromboembolic events are quite frequent as a result of long-term consequences of COVID. So a study that evaluates 150,000 patients that survived COVID demonstrated that thrombotic disorders and poor cardiovascular outcomes were between the most frequent consequences of COVID-19, increasing the burden of disease one after the onset of COVID, especially in patients that had severe forms of disease. So there are quite a number of open questions regarding the long-term consequences of COVID. What's the occurrence mechanism, how we define disease? Most of the symptoms are quite unspecific. How can we determine biomarkers and therapeutic approach for these long-term consequences of COVID? And also, there are quite a number of open questions on what are the mechanisms and the targets for long COVID infection. So we studied 50 COVID-19 pneumonia survivors that after 60 days of the onset of disease up to 180 days. We are now closing up the data for 2-year follow-up of those patients. Those patients had moderate pneumonia, only a small percentage had severe pneumonia, and only 10% requiring invasive mechanical ventilation. Important to say that all patients report at least one persistent symptoms, and 41% reported more than 6 symptoms after the onset of COVID. The most frequent reported symptom was the respiratory symptoms, including shortness of breath, difficulty remembering, and poor concentration, anxiety, depression were also very often reported. So we first asked, would those patients exhibit persistent lung immune and metabolic activity after COVID? So here we can appreciate the CT from the patient within the acute phase, and 60 days after disease, we repeated the exams and had the PET-CT performed. And as we can observe here, those patients, they exhibit an increased immune metabolic activity, both in the parenchyma interstitium of those patients. So we went on to ask, would that be elevated biomarkers of endothelial cell activation, coagulation, and inflammation present on those survivors of COVID-19? And that is what we can appreciate here, that there are indeed increased markers of endothelial cell activation, as can be observed by angiopoietin increased levels, as well as increased levels of VCAM, soluble VCAM and soluble ECAM in the plasma of those patients. Inflammatory markers, including IL-6, were increased, and also coagulation activation marker, D-dimer, were also persistently increased in the plasma of those patients. So we went on to ask, would platelets be activated? Could that contribute to the disease? So first we investigated the activation of platelets in those patients within more than 60 days after COVID. And we can see here that indeed platelets from those patients exhibit signs of platelet activation by measured both by the expression of P-selectin and CD63 on the membrane of those platelets, demonstrating release of alpha and dense granules by those platelets, as well as when we get the platelets from those survivors of COVID and put them ex vivo without stimulation, we can see that platelets from survivors of COVID exhibit much higher inflammatory mediator secretion when compared to platelets from healthy donors. And that could be observed for PDGF and Rantas. We went on to see what would, if we could see markers of platelet activation in the plasma of those patients. And again, we see quite increased soluble P-selectin, PDGF increased, PF4 increase, and also thromboxane B2 increase in the plasma of those patients. Platelets from the COVID-19 survivors, they not only exhibit signs of activation, but they also are hyperreactive to a second stimuli, and that's what we can observe here. First, in assay of platelet adhesion upon fibrinogen-coated slides, we can appreciate here that platelets from survivors of COVID are much more active in those spreading and adhesion to fibrinogen. And also, when we use suboptimal doses of thrombin, we can see that thrombin in very low doses can activate platelets from COVID-19 survivors, but not from healthy donors. And when we use optimal doses of thrombin, this activation is much, much higher in the COVID-19 survivors, indicating that platelets are hyperreactive upon stimulation. In addition, we also see increased levels of circulating tissue factor as well as microparticles, mostly platelet microparticles expressing tissue factor upon COVID-19 survivors. So we used an ex vivo assay by using plasma from the COVID-19 survivors, stimulating platelets from healthy donors to gain insights on what were the mechanisms involved in those activation. And as we can appreciate here, these mechanisms involve different signaling pathways, including mechanisms that are partially dependent on cyclooxygenase, so it can be inhibited by aspirin, as well as pathways that involve P2X7 receptors and integrin receptors as well. So, that comes to our current view of the mechanisms of thromboinflammation in the long COVID. We see that indeed we see signs of persistent endothelial cell activation, signs of increased expression of tissue factor, which could contribute to the hypercoagulable state on those patients. Platelet exhibit persistent activation and hyperreactivity, and that are both influenced by the inflammatory milieu of those patients, as well that enhances the inflammatory response in a two-way pathway. And now there are persistent open questions that involving what are the triggers to maintain this persistent activation. Some recent data indicates that there are viral reservoirs that could maintain this proactivation status. There are hypotheses of the presence of autoantibodies, as well as our group has previously demonstrated that COVID can enhance endogenous viral elements, mostly HERV-K mRNA, and that associates with the thromboinflammatory response in the COVID-19 patients and can potentially participate in those mechanisms here in the long COVID. And there is quite an open questions on how those mechanisms do indeed contribute to the signs, symptoms of long COVID. So as a take-home message, post-COVID syndrome is frequent. It is quite still variable according to the patients. The incidence varies between non-hospitalized and hospitalized cases. Anyway, due to the high number of patients with COVID, it's a very important task ahead that we have in terms of number of patients that may have long COVID. And what I would like to add to you today is that the pathogenesis of post-COVID syndrome is multifactorial, and potentially there are different phenotypes of patients with long COVID, and at least part of those patients includes a persistent thromboinflammatory response. So I'd like to finish by acknowledging the many collaborators in this study. The part of platelets in post-COVID was mostly performed by a very talented PhD student in the lab, Remy Martins Gonçalves, and involved the collaboration with different groups in fiocruz and at the Federal University. And I will be very happy to take any questions.
Thank you, Patricia. The floor is open for questions. How do we do that? Is there, okay.
Go to the
I
mic.
think, yeah.
Thank you. Thank you very much. This was really a scholar presentation, Patricia. Fantastic. Congratulations. Really major quality of work and large amount of work. I have sorry, Mauro Perretti, Queen Mary University of London. I have a couple of questions. My understanding that normally platelet leukocyte aggregates are transient. You know, they can aggregate and they can detach. So one question for you is whether the higher aggregates you found in COVID patients could be due not only perhaps by a higher adhesion, but also by a lack of the detachment process, whatever that is mediated by.
Yeah, it is possible that both mechanisms act together. So indeed, they are very dynamic in those interactions, and to ask those and to address the platelet-leukocyte interaction, we use fresh blood and immediately used to access those interactions because those are lab interactions, but we can indeed modulate it by the adhesion. So P-selectin and alpha-2B are two of the mechanisms that are very important, both in platelet leukocyte, platelet monocyte and neutrophil interaction. But it is possible that the lack of dissociation does also play a role.
Question? Very elegant studies when you added platelets from COVID to normal monocytes and vice versa. So is the system pharmacologically susceptible? If you add a platelet inhibitor, can you then downregulate the production of cytokines and other factors? Like an ADP antagonist, I think.
Yeah. It is, so the system, and that's, so to modulate pharmacologically, we've been using those systems of interaction as well as using plasma from the donors to activate the platelets. In the case of platelet, the answer is not that easy because sometimes some of the platelet, direct platelet agents are not that efficient to inhibit the response, the immunophenotype change in the monocyte. In part, partially our view is that because we get platelets that are already activated, so blocking some of the pathways of platelet activation are not enough to overcome the platelet that already come activated.
Thank you, Mauro.
The final question, because it's a very exciting data set you presented. I must say those experiments at the end in long COVID with the plasma were, you know, they were convincing, but up to a point. So my question is, do you think there is a defect in the megakaryocyte in the long COVID patients, and perhaps the virus actually, that you said, the reservoir is in the bone marrow?
Yeah, that's part of our hypothesis. It's now became clear that megakaryocytes resident of the lung there are quite a number of megakaryocytes that are resident at the lung. We believe that those megakaryocytes may maintain some of viral reservoir or viral particles or components that can maintain the stimulation and release pre-activated or immunophenotype differently platelets, but that's still an open part. And I agree with you, it's quite hard still to work with the long COVID. It's quite a confusing scenario, mostly now at the point that we are on the pandemic, that most of the patients had different courses of COVID-19 disease and also different courses of infection. The patients that I showed to you, it was before vaccination, so before the starting of vaccination. So those were a more select group of patients that were included, and we could follow them up for 2 years, and we had the chance to compare back them with themselves. So that helps to get something of the confusion out, but I agree with you that it's still very hard to get a good sense of the syndrome.
Thank you, Patricia.
Thank you.
Okay, so I would like to invite to the floor Christina Thomson. She's from the University of Aberdeen, and her abstract was selected from the pool of abstracts sent. And she's going to be talking about FPR2 in the context of bacterial infection. Thank you, Christina.
Thank you. Good morning. So as I was introduced as a PhD student at the University of Aberdeen, but this work today was completed a year ago when I completed my Masters of Research in Dr. Don Thompson's lab. So I'll firstly introduce this formal peptide receptor 2. So it's a receptor found in humans. It's found on the surface of a multitude of different cell types, but most notably on immune cell surface. It has a diverse range of ligands, making it highly targetable, and these include small molecules, peptides, and proteins. But really importantly for the inflammatory response, FPR2 has been identified as the master switch between the pro-inflammatory phase and the pro-resolving phase of inflammation. Studies have then further highlighted this role, with FPR2 knockout mice failing to recover from severe infection and sepsis. This was then further confirmed with the annexin knockout model, which displays this same phenotype, with annexin here being a ligand of FPR2. So FPR2 is also a GPCR, and GPCRs are very important in pharmacology. They're highly druggable, highly targetable, and they make up the single biggest family of drug targets alone. FPR2 is also GI coupled, meaning its activation inhibits the activity of cyclic AMP. So knowing all this, we just wanted to determine how activation of FPR2 could be beneficial to the inflammatory response to treat infection. To look at this, we chose two different infection strains, gram-positive and gram-negative. So we've got the gram-negative component, LPS, derived from E. coli, and the gram-positive component, peptidoglycan, derived from Staph aureus. And we wanted to determine whether there would be differences between these two strains. So having picked these two strains, we then needed to find a pathway to effectively compare infection. So with the gram-positive peptidoglycan being recognized by the TLR2 and the gram-negative LPS being recognized by the TLR4, we then needed to find a pathway where we have this convergence. And this occurs on the NF-kappa B pathway, which is a really important mediator of the inflammatory response. So upon TLR signaling and recognition of pathogen bacteria, we have activation of these IKKs, which then lead to the phosphorylation and activation of this NF-kappa B complex. So this contains an inactive NF-kappa B bound to an inhibitory I-kappa B. This I-kappa B is then rapidly phosphorylated and then degraded, which allows the NF-kappa B to become activated and translocate to the nucleus in order to promote this inflammatory response. So knowing this, we then also want to determine whether FPR2 could influence this and how it may be doing so. So to do this, we made use of the raw cell line, which is a mouse macrophage-like cell line. We stimulated with our 2 different infection strains, either LPS or peptidoglycan, and used Western blotting to observe acute NF-kappa B signaling. We've then introduced agonism of FPR2 with this W peptide ligand, which is a potent FPR2 agonist. And then we've also had a wee look at overexpression of FPR2, which I'll explain a bit later. So, as I say initially, with these raw cells, we just wanted to compare our 2 infection strains and see whether they could activate this NF-kappa B pathway comparably. And we found that they did so. So with phosphorylation of this NF-kappa B, we found that this began within 5 minutes with both peptidoglycan and LPS. It was sustained then following a peak at around 15 minutes for the remainder of the time course. And as I say, this was the same between LPS and peptidoglycan. We then wanted to look at this inhibitory protein that is rapidly phosphorylated and degraded. So with phosphorylation between the 2 infection strains, we found that this began at 5 minutes as before with the NF-kappa B, same for peptidoglycan here. Slightly decreased at around 15 minutes, and then rapidly increased again, and we saw maximal around the 1 to 3 hour time point. Again, the same for both LPS and peptidoglycan. We then looked at total protein expression of this I-kappa B protein. So from the beginning of the time course, prior to stimulation, we have expression here. It then drops slightly and then completely degrades around the 15-minute time point. Again, the same between LPS and peptidoglycan, before then rapidly recovering and peaking at maximal around 1 to 3 hours. So having identified these 2 infection strains and they elicit a comparable response of the NF-kappa B pathway, we've then introduced agonism of FPR2. So we repeated our time course, but with an added treatment group, treated for 2 hours prior to infection with this W peptide ligand. So with phosphorylation of NF-kappa B, we found no changes. We found it elicited the same response between LPS, peptidoglycan, and adding agonist. However, when we added agonist and looked at our inhibitory, I-kappa B alpha, we found an interesting result. So with phosphorylation, I'll go back. With phosphorylation, it remained the same between treatment groups. However, with total protein expression, we found something really interesting. So we have this rapid degradation around the 15-minute time point. However, in LPS-exposed cells, we found that this recovery at the 1 to 3 hour time point was significantly reduced. However, when we compared this to peptidoglycan, we found the opposite trend and found that it actually resulted in increased recovery. So FPRs and GPCRs in general are upregulated on demand following infection, and we found in our raw cells stimulated with LPS that we found upregulation of the FPRs around the 2 hour time point, but even more so around the 4 and 6 hours. However, we can just note here that there's no acute signaling of FPR or FPR2 in these raw cells following agonist treatment for 5 or 20 minutes. So to overcome this, and no longer having to wait for the endogenous upregulation of the receptor, we created a model system. So we now have these same raw cells infected with a lentivirus that had the N-terminally flag-tagged FPR2, meaning that they can overexpress this receptor. We then tested this using microscopy. So our unstimulated cells, the receptor is expressed on the surface here, just as it would be in a model stable expressing HEC cell. And then following 30 minutes of agonist treatment, we found that there was internalization of the receptor, again, as we would expect in a model HEC system, but now in an immune cell. We then tested this against our acute treatment with AKT signaling, and with overexpression, found that 5 minutes of agonist treatment resulted in AKT phosphorylation. So now, with our model system, we went back, repeated our time course, and compared our plain raw cells with our overexpressed raw cells. So there's no agonist here, but just repeated as before. Here we found a slightly more exaggerated response. We found that overexpression of FPR2 resulted in increased NF-kappa B phosphorylation following stimulation, and this was the same for both LPS and peptidoglycan. We then went back to look at our inhibitory, I-kappa B, and found that the overexpression of FPR2 mimics this agonism that we saw previously with the W peptide ligand. We have reduced recovery in the LPS-stimulated cells, but increased recovery in the peptidoglycan-stimulated cells. So really, just to summarize that data there, we saw that LPS and peptidoglycan elicited a comparable response in the activation of NF-kappa B. However, when we introduce agonism and activation of FPR2, we see an alteration in the response. So we saw that LPS exposure resulted in a significantly reduced recovery of I-kappa B following degradation. However, the opposite was observed in the peptidoglycan, so the gram-positive stimulated cells. Then our overexpression model mimicked this same response in this agonism. So how can we relate this all back to FPR2? Well, studies have highlighted AKT as a key regulator of the NF-kappa B pathway. And it's also known that FPR2 signals downstream through AKT upon activation. So we've hypothesized here that FPR2 could be influencing this NF-kappa B pathway via AKT signaling. So just to conclude, FPR2 activation influences the NF-kappa B response, but depending on the infection strain. So with the gram-negative LPS, we observed a blunted I-kappa B alpha recovery, which potentially could lead to enhanced NF-kappa B signaling. However, the same wasn't observed in the peptidoglycan, but we had this enhanced I-kappa B alpha recovery. So in conclusion, FPR2 could prove useful as a therapeutic against severe infection, but in combination with current antimicrobial therapies. Thank you. I just want to also thank my supervisory team, Dawn and James, and my lab, pictured here, and also my funders. So Friends of Anchor funded the project for my master's, and then the BSI and the BPS allowed me travel grants to be here this week. Thank you.
Thank you, Christina. Any questions from the audience, please? In principle, activation of NF-kappa B is actually useful for the animal or the host to deal with the infection. Mm-hmm. So did you actually try infecting with the bacteria itself or giving the bacteria to these, and whether you would change with your agonist or your cell line? Given the bacteria, not necessarily the LPS.
No, so we didn't try that. I'm not sure how that would affect that. I don't know how that would maybe work in our cell line as well, if we're having to then put them back into the incubator if they were infected per se. I'm not too sure.
Thank you. Yes, over there.
Mic 4?
You can
Mic
start
4?
there, I suppose. And then
Quick question, great talk, super stoked on this receptor actually. I want to get some clarity. When you overexpress the receptor, mm-hmm. I don't recall seeing any increase in the phospho-AKT signal under resting conditions, like in the absence of the W peptide stimulation. Is that correct?
Yes.
Okay, so you didn't see any phospho-AKT signaling when you didn't have stimulation, yet you saw an effect on NF-kappa B signaling in the absence of stimulation in your overexpression system. Mm-hmm. What's happening there?
That's it. I've never actually really thought about that. That's really interesting. I mean, I guess, although it's overexpressed, it might not be activated until the W peptide ligand has activated it, I suspect.
I think there's a question over here, please. Mm-hmm. Yeah, go, Max.
Thank you. That's it. Yeah. Did you ever check if your macrophages changed the phagocytosis index while treating with this inhibitor?
We haven't looked at that at all. There was a long to-do list last year when I started my master's, and one of the things was to get to then look at phagocytosis and cytokine release and things like that, and we just still haven't got around to it. So.
Okay, thank you.
Very nice talk. This FPR2 receptor has this kind of bias, can be activated by pro and anti-inflammatory ligands. Have you tried to compare that sort of activation, pro and anti-inflammatory, using your system? The repercussion in terms of
with like maybe different ligands depending on what they induce. We haven't tried that. We've just been using this W peptide. It's a great tool in the lab, but then when we look at ligands that would be more appropriate for therapies, sometimes they don't work as well as this W peptide. So the W peptide is an ideal lab tool, but the FPR2 ligands, there's a lot of differences between them, and maybe they activate certain things and not others.
Thank you.
Thank you, Christina.
Thank you.
Okay, our next speaker will be Dr. Maximiliano Ferrero. Max is graduated in biotechnology at the National University of Quilmes, Argentina, and obtained a PhD from the University of Buenos Aires in 2014, working in immune parasitology and biochemistry. During his postdoctoral training at the Oswaldo Cruz Foundation in Rio de Janeiro, Brazil, he developed prospective studies of drug repurposing and drug development for treating exacerbation of pulmonary inflammation caused by viral infection and tabagism. Since 2021, he has worked at the Max Planck Institute for Lung and Heart, cooperating with the Biomedicine Research Institute of Buenos Aires and the Justus Liebig University of Gießen, Germany, exploring inflammation resolution pathways and lung tissue regeneration to improve lung function during severe pneumonia. The title of his talk is Neutrophils and Cigarette Smoke-Induced Exacerbation of Lung Viral Infection.
Okay, thanks so much for this kind introduction. Good morning, everyone. In the next 20 minutes, I'm going to present some of the results obtained in the laboratory of inflammation at Fiocruz Institution in Brazil. This is entitled, as Marco already says, Neutrophil and Cigarette Smoke-Induced Exacerbation of Lung Viral Infection and Modeling COPD Crisis. The principal aim of this project was to establish a model of acute inflammatory exacerbation in lungs, difficult to treat, that resembles COPD exacerbations to use as a platform for the discovery of novel therapies. But why did we want to study COPD exacerbations? Well, COPD stands for chronic obstructive pulmonary disease, and some key facts are this is the third leading cause of death worldwide, and cigarette smoke represents a major risk factor, but also airway pollution is a major risk factor. What happens is that sustained exposure to noxious particles and also microorganisms in the air can trigger chronic inflammatory response in the airways that is characterized by neutrophil activity. And this triggers, of course, acute inflammation, but also chronic airway inflammation, tissue damage, and remodeling, often seen as emphysema, which is the breakage of the alveolar wall, but also remodeling of the airways, like seen in small airway disease or chronic bronchitis. Altogether, this contributes to limit the lung function. But moreover, COPD patients are exposed to respiratory infections. They are particularly vulnerable to respiratory infections indeed, and these infections can trigger COPD crisis or acute exacerbations of COPD. These are the main cause of hospitalization, morbidity, and mortality of the disease. And for instance, in this example, influenza virus, which is often related to COPD crisis, would represent like an additional inflammatory stimulus to an already inflammatory background, triggering the expression of further inflammatory mediators like IL-8, CCL3, MCP1, IL-6, TNF-alpha, which would recruit further neutrophils to the tissue, principally via CXCR1 and 2 chemokine receptors. I will go back to this point a few slides ahead. And all these inflammatory background would, of course, increase oxidative damage, and clinically, this would convert into sudden worsening of COPD symptoms, accelerated lung function deterioration, disease progression, and also increased serum IL-6 levels, which normally correlate with the worst outcome. For instance, in this example, we have how the probability of surviving of COPD patients is associated with the number of exacerbations this patient presents. In group A, we have patients that had no exacerbation in a year. In group B, we have patients that experienced 1 or 2 exacerbations in a year. And in group C, 3 or more exacerbations in a year. The problem is that there are no specific treatments to control COPD exacerbations, and most COPD patients are not sensible to corticosteroid treatments. So, in order to accomplish our aims, we need to reproduce in our mice model the exacerbated neutrophil response and inflammatory mediators, the lung function impairment, oxidative stress, of course, and systemic inflammation and reduced survival. And how did we do that? Well, we had a 12-day setup in which we exposed mice to 11 days cigarette smoke exposure. And on day 7, we infect those mice with this mouse-adapted influenza virus strain, H1N1-PR8, in 2 different doses, 100 PFU and 1,000 PFU. And of course, we had 3 groups of control mice, the mice that were only infected, mice that were just exposed to cigarette smoke, and also mice exposed to room air. By day 12, the readout would be weight loss, lung function, leukocyte infiltration in the airways, inflammatory markers in lung, oxidative stress, and serum IL-6. In our first approach, what we found was that only the mice that were infected with 1,000 PFU, regardless if they were exposed or not, lost significant amount of weight. When checking the airways, we saw that 1,000 PFU virus dose increased neutrophils in the airway, nor the cigarette smoke exposure did that, or 100 PFU virus also did not do that. But what was interesting to find is that when mice were exposed to cigarette smoke and then infected, there was a clear exacerbation of neutrophil recruitment to the airways. And this was further confirmed for lung tissue, measuring MPO activity. So from now on, I will continue just with 1,000 PFU, as with 100 PFU, we did not see any of the change that we wanted to see. For instance, we also quantified some inflammatory mediators in lung tissue, like cytokines and chemokines, often observed in COPD patients while experiencing crisis. We had TNF-alpha, which was significantly increased in mice when they were exposed and infected. IL-6 was increased in infection, but exacerbated when mice were exposed and infected. None of those increased when mice were just exposed to cigarette smoke. And some chemokines like KC, which resembles IL-8, is like the mouse version of IL-8. It was increased in infection, however, it was exacerbated when mice were exposed and infected. Also CCL3 and MCP1, same scenario. But differently, Rontes, which is a chemokine associated with lymphocyte and eosinophil recruitment, we didn't saw any difference between mice that were infected with 1,000 PFU, regardless if they were exposed or not. Indeed, we did not see any difference in the number of lymphocytes that were recruited to the airways. This is coherent with Rontes. When checking at lung tissue sections, we saw again, it was a huge inflammatory exudated seen in mice that were exposed and infected compared to mice that were just infected, or even to mice that were just exposed. And indeed, we saw some signs of lung tissue remodeling that looked like emphysema in those mice. Going further, in oxidative damage, we quantified lipid peroxidations in lung tissue and found out that just the mice that were exposed and infected had significantly increased lipid peroxidation in lung. And this could be related, or we think that this might be related, to the fact that viral infection decreases catalase activity in lung tissue. Then we checked systemic inflammation by quantifying serum IL-6 levels in mice. We saw a tendency to increase in mice that were infected, but clearly there was a significant increasement in serum IL-6 level when mice were exposed and infected. Further, we went to evaluate lung function by whole body plethysmography for non-invasive measurement. The advantage of this technique is that we could record lung function in mice that were awake along all the experiment. This gives you the value of pNH, which correlates with airway resistance. What we saw is that mice exposed to cigarette smoke didn't show any difference in pNH value across the experiment, but when infected mice show a progressive increasement of pNH values. However, when mice were exposed and infected, there was an exacerbation of this condition. We wanted to confirm if indeed what we were seeing was increasement of airway resistance. We used the day 11, where the gap between infected and exposed and infected mice was higher, and indeed found that when mice were exposed and infected, there was a significant increase in airway resistance. All these changes were not apparently related to a different amount of virus in the lung tissue, as both infected and exposed and infected mice showed similar viral titers. In terms of antiviral response, we quantified gene expression of type 1 interference and type 1 interferon-associated genes, and found out that after infection, there was an increase, of course, in these genes, but when combined, cigarette smoke exposure and infection, there was a decrease in the antiviral response. So these might be a kind of different inflammatory process that both stimulus trigger. We checked dexamethasone resistance because dexamethasone is the main therapeutical treatment COPD patients receive as anti-inflammatories when experiencing crisis. We treat our mice 24 hours after infection, once a day for 4 consecutive days with 1 milligram/kilogram via oral, and found out that dexamethasone treatment could not revert the impaired lung function that these mice presented, neither the excessive recruitment of neutrophils to the airways, nor in the lung tissue. But most importantly, we could see that upon infection, the survival rate of the mice was dramatically reduced to 60%. However, this condition was exacerbated when mice were exposed and infected. And dexamethasone could not prevent this exacerbation of mice mortality. So by the moment, we had in our model exacerbated neutrophil response and inflammatory markers, also lung function impairment, increased oxidative damage, systemic inflammation, and decreased survival. But overall, we had a difficult to treat model, and it's also a short-term model. And this allowed us to treat many different drug candidates, or indeed 3 drugs that are prescribed for COPD patients. One is roflumilast, which is an anti-inflammatory, and 2 of those, ipratropium and tiotropium, are bronchodilators. None of those drugs could prevent mice for the deterioration of lung function, neither to reduce this exacerbated recruitment of leukocytes to the airways. But in the last half of my talk, let me tell you about 2 successful stories. This involves two chemokine receptor antagonists. As you may know, chemokine receptors are those who drive leukocyte migration across gradients of chemokines. The first target we wanted to check at, of course, was neutrophilic inflammation, as neutrophils are associated to COPD and acute exacerbation of COPD pathology, and also because neutrophils are recognized as main contributors to corticosteroid resistance. Moreover, in our model, we saw that lung function impairment was correlating with the increase of neutrophils in the airway. And this is also observed in the clinic in COPD patients while experiencing crisis. So we choose lalorexine, which is a novel compound, a non-competitive dual allosteric inhibitor of CXCR1 and CXCR2 receptors. These 2 chemokine receptors are very, very related to neutrophil migration. Lalorexine proved to be safe in phase 2 clinical trials, and there are 2 previous reports which proved its efficacy in preclinical models of arthritis. So we choose a therapeutic setup in which we start the treatment with lalorexine 48 hours after the infection, once a day for 3 days in a row, with 10 milligrams kilogram orally, and found out that indeed lalorexine could reduce the leukocyte numbers in the airways of mice treated compared to the mice that were exposed and infected, of course, particularly by decreasing the number of macrophages, but most importantly, by decreasing the number of neutrophils that these mice presented in the airways. No alteration was seen in the number of lymphocytes in the airways of mice, of the infected, exposed and infected, and exposed, infected, and treated, of course. And similar situation we observed in the lung tissue of these mice regarding the neutrophil activity. So ladorexine reduced neutrophil activity in lung tissue of mice when treated. We could also observe ladorexine anti-inflammatory effect in lung sections compared to the mice that were untreated. Plus, we saw that ladorexine prevented the excessive lipid peroxidation in lung tissue, but also prevented the increased values of IL-6 in serum. Further, we went to check if these changes correlated with mice outcome and found out that indeed ladorexine prevented from the first day of administration the impairment of lung function that mice that were exposed and infected presented. And these benefits were conserved along the experiment. Finally, and luckily, we found out that ladorexine could prevent the increased mortality that mice exposed and infected presented. All these changes that we found with ladorexine did not relate to viral titers, but indeed ladorexine decreased significantly the weight loss of those mice. So with ladorexine therapeutic treatment, we could protect mice from exacerbated neutrophil response, improve lung function, protect from oxidative damage, and also protect from systemic inflammation and decrease survival. Next. Good. So the second story is about maraviroc. Maraviroc is a drug that targets CCR5. CCR5 is a chemical receptor again. We use maraviroc for two main things. One is because it's an already approved drug for human treatment, but also because CCR5 expression is augmented in COPD patients. Moreover, we found out that in our model, CCL3, which is a chemokine that can bind CCR5, it's exacerbated in mice when they were exposed and infected. And this is also observed in the clinic in COPD patients while experiencing crisis. So in our lung macrophages and lung neutrophils, CCR5 is also expressed. So we had a similar setup to that one used in ladorexine case. We started 48 hours after infection with the treatment with maraviroc, once a day for 3 days in a row with 10 milligram kilogram per oral route, and find out that indeed maraviroc could reduce the number of leukocytes in the airways of mice treated and also prevent from lung function impairment. However, a CCR1 antagonist, which is other chemokine receptor that CCL3 can bind, did not present any effect on these parameters that we evaluated. So we believe that at least in our model, CCR5 is kind of contributing to our inflammatory response. More in detail, we checked macrophage number, neutrophils number, and lymphocyte numbers in the treatment with maraviroc and found out that indeed there was a nice decrease in the macrophages number in the airways of the mice that were exposed and infected. Some significant decrease in neutrophil numbers, and lymphocyte remained the same among all those groups. In lung tissue, by flow cytometry, we found similar results, indicating that number of macrophages and neutrophils are decreased upon maraviroc treatment. Finally, serum IL-6 level that were exacerbated in mice that when exposed and infected, the level of IL-6 were reduced in maraviroc treatment also, but most importantly, maraviroc could prevent from the exacerbated death rate, while viral titers of all infected mice remain the same. So with maraviroc therapeutic treatment, we could protect from exacerbated leukocyte recruitment, improve lung function, protect from systemic inflammation, improving mice survival. Some concluding remarks that I have is that we could establish a short-term model of acute inflammatory exacerbation, steroid-resistant, in lungs that resembles COPD exacerbation, which could be used for uncover novel therapeutic targets and evaluation of drug candidates. Chemokine receptors may be good candidates to control lung inflammatory exacerbations in COPD patients while experiencing COPD crisis, as we found out that CCR2 and 1 antagonism could modulate the inflammatory exacerbation in lung of cigarette smoke-exposed and infected mice and improve mice outcome, and also find out that CCR5 chemokine receptor inhibition using a drug repurposing strategy could control the inflammatory exacerbation and improve mice outcome. With that, I want to thanks a lot for your attention, and thanks, of course, to the Laboratory of Inflammation where I did this work, principally to the head of the laboratory, Marco Aurelio Martins, thanks a lot, and our collaborators in the Laboratory of Respiratory Virus and Measles at fiocruz also, particularly to Professor Cristina Couto Garcia, and of course, to our financial support agencies. Thanks a lot. I'm open for any questions you want to make.
Very nice presentation. The paper is open for question, if you have. Okay.
Yeah. Richard Amison from King's College London. That's a really nice presentation. I was just got a quick question of whether you've considered, given the role that we know that platelets play in a lot of acute infections, whether that's bacterial, fungal, or viral, and particularly in sepsis, their association with worse prognosis. I was wondering if you've looked at any of the platelet function or activation states, or even circulating platelet numbers in your model.
Sorry. Do you mean if I checked out blood situation?
Yeah, I was just wondering in the validation of your model in the first section. I was just wondering if you've looked at any platelet function or activity in that model.
Not platelets, not. Sorry. We found out that in blood, for instance, in our model, we saw a decrease in inflammatory cells. Inflammatory cells. When you expose mice to cigarette smoke, normally you see a decrease in leukocytes that are circulating. While when you infect those mice with influenza, you saw an increase. The result of the interaction of these two factors, at least in our model, was a kind of average between those two effects. So in terms of blood, it was interesting to see that while IL-6 was increased, there was not really a change in the number of leukocytes in the blood. But this is all I can say. We didn't went through evaluating platelets or any other marker in blood.
And then just quickly as a follow-up, have you looked at any, because obviously you've got that systemic inflammation present and the increased mortality. Mm-hmm. Have you looked at any systemic dissemination of any of the viral particles?
Yeah, we did indeed, and we find much more virus in the spleen of those mice. We don't know if this can correlate to alveolar damage and leakage of this virus. We don't know if this virus was alive there, but indeed there was more virus in the spleen, and I cannot explain you why, but we found out that. Yeah.
Really nice presentation. Thank you.
Thank you.
Hello, hi, it's Jamie Chen from Queen Mary University of London. Very brilliant talk, very nice research on drug repurposing. I was wondering, because your mouse model is a short-term model which lasts only for 12 days, have you ever thought about monitoring these mice for longer term, for example, by quantifying the lung fibrosis and the lung compliance? Yeah, excellent point. The thing is that in our model, it's so severe that mice do not survive. It's good to study the drug candidates because it's a severe model. If a drug works there, it might be interesting. But the problem is that we cannot do a follow-up. So what we planned before the COVID pandemics, actually, it was to reduce the viral doses to see if we could evaluate the mice further. Indeed, to see if we could find, for instance, emphysema or, of course, lung fibrosis, and study long-term changes in these setups. But the thing is that at least in this model, we could not follow up the mice because they do very wrong. Thank you.
If no more questions, I think we can move on to Mauro.
Thank you.
Thank you very much, Max. So our last speaker of this morning is Professor Mauro Martins Teixeira. Mauro Teixeira studied medicine at the Federal University of Minas Gerais, after which he began his postgraduate studies at the Lung and Heart Institute, Imperial College London. He then returned to Brazil and is a full professor of immunology at the Department of Biochemistry and Immunology of the University of Minas Gerais. Mauro is a Brazilian Academy of Science member and principal investigator of the National Institute of Science and Technology in dengue. Developing anti-inflammatory drugs for infectious disease is one of his primary scientific interests. I think Mauro is increasingly trying to combine his expertise as immunopharmacologist and clinician, developing and testing novel medications and treating patients suffering from COVID and other disorders. The title of his talk today is Resolution of Inflammation in Infectious Disease.
So thank you, Marco. Thank you, everyone, for staying on. And thanks for IOFAR for accepting our indication for this session. So I will talk to you about the context of infection and inflammation, especially arboviral infection. So how does that, okay, good. I will try to go over 4 points over this presentation. You know, the relevance of arboviral infection, especially dengue, it's considered a neglected disease. So not many people know about it. The concept of anti-inflammatory drugs, or what we call host-direct therapies as adjuvant for infectious disease in general. The concept of resolution of inflammation and whether that's applicable to infectious diseases. Okay, so we use the word arboviruses or arboviral infection as a short form of arthropod-borne viruses. This is a really huge range of diseases which have this characteristic of actually having an arthropod that will transmit disease to humans. I think they are increasingly important simply because the vectors are increasing everywhere. With changes in temperatures, things are moving up north. You know, for us that do research, that's actually not bad because it becomes more important. So as we infect Europeans, things become more important. We get actually able to do some research and actually eventually find the treatments. This is sad, but it's true. And, you know, these are the most important arboviral infections, but there's much more to that. And the risk is absolutely increasing worldwide, you know, and WHO is using terminology such as One Health, actually to try to actually study diseases in which there is a huge and important human component, but if you don't change and if you don't address the environment, you will not get rid of these diseases. As you can see, you know, you find these diseases everywhere where humans are. Sorry, Canada, you know, lots of parts, it's not, you can't live there. So whenever there is evidence of human settlement, there will be evidence of an arboviral infection. You know, I don't know why Alaska is out of here, but it's probably too cold for the vectors to live there. And again, you know, a range of different diseases, different virus classes, you know, from dengue to the flavivirus to a lot of the encephalitic viruses as well. Of these, dengue is for sure the most important arboviral infection. There are about half of the population of the world at risk. About 10% of people are infected yearly, so that's about 400 million infections yearly, and of which about 20% are clinically apparent. And, you know, COVID actually made everything a little bit relative. You know, dengue was considered a lethal disease, but in fact, it only, inverted comma, kills about 0.1, 0.2% of people that are infected. But as you can see with these huge numbers, actually it has a huge burden on places where it comes. And very important, as you can see here from 43 to 2013, the places which are now infected with dengue have increased a lot. And the other thing that we're finding is that more different types of dengues are everywhere. Now from dengue 1 to 4, it's everywhere, you know, especially in the tropical regions of the world. because there are no treatments. Vaccines are becoming available as we speak. There is a new recent vaccine approved by Takeda. There is one by Sanofi. Butantan has a clinical trial. There are certain vector-based strategies that are being approved and being tested. Now, there are no treatments as yet. So DNDi, or Drugs for Neglected Disease Initiative, has created a Dengue Alliance, which is mostly a South-South collaboration in different parts of the world, Thailand, India, Malaysia, and Brazil, which has the objective of actually generating new drugs to treat dengue. So the overall strategy is try to develop two kinds of strategies, pretty much as we have done for COVID. One is focused at what we call direct-acting antiviral agents. So these are direct antiviral. Whether they act on the viral proteins or host proteins is something different, but they are aimed at the virus, or host-directed therapies. And similarly to COVID, dengue has this very unique clinical presentation. So in the first 3-4 days of the diseases, patients are incredibly ill. They have a lot of fever, and they seek the doctors very quickly. Now, that's actually, although the patients are really feeling very bad, that's actually the safest part of the disease. As fever comes down, the immune system kicks in, and so there is a huge vascular permeability in most patients. Now, in 10 to 20% of them, that is clinically significant. In not so many, about 5% will eventually need medical support, and death occurs in these individuals. Although we use terms such as dengue hemorrhagic shock or dengue shock syndrome, so dengue is what we see as absolutely a viral shock syndrome, right? So as you have sepsis, bacterial sepsis, you have a viral sepsis, which is dengue. It is characterized by vascular permeability, a lot of endothelial damage, a lot of platelet changes, as Patricia showed. She has some beautiful work on that as well. And actually, that helped a lot on the COVID studies. We were discussing about that. So it is in these critical phases when people die, they need a lot of medical attention, they are taken to hospital. You know, I haven't seen, but in places like my city, we have dengue almost every 3 years. So, because there are four dengue serotypes, they keep changing each other. When there is a massive dengue, it protects indistinctly against the other serotypes in the first 2 years. And then another serotype kicks in, and there is another massive epidemic. So the metropolitan area is about 5 million. In any particular epidemic, you have about 400,000 cases in 2 months. You can imagine what it does to health systems. It just collapses, right? So you need a way to actually keep people at home, prevent them from going to the hospital, and keeping them safely at home. So one of the other options is try to develop what is called host-direct therapies. I will go into that. As far as the direct antiviral drugs, there are different ways of actually developing drugs. So I will just show our work, but there are lots of people doing work around that. There are drugs that actually bind to the NS5 or the RNA-dependent RNA polymerase. Inhibitors of that enzyme actually are very effective inhibitors of dengue replication, and they actually may be useful in vivo. You may actually interact with different virus proteins, such as NS3 and F5 interaction. And it's interesting that these drugs are very broad spectrum, so they will actually work against a range of flavivirus, including Zika and yellow fever, for example. So there is actually interest in developing these kind of compounds because at presentation, dengue is totally indistinguishable from Zika, chikungunya, or any of the other acute arboviral infections. Now, of the actual acting antiviral drugs, there are 3. They are more advanced in clinical development. There is this compound from Atia. It's a guanosine nucleotide analog. So it will prevent elongation of the RNA. The program has been halted recently because the company actually found out that it was too expensive to develop a drug for dengue. which is interesting. 4 billion people are affected, but it's expensive to develop a drug. In actual terms, it is, because you need a very large clinical trial. And actually, the money for that sort of clinical development is not easy to find. Janssen has a compound. It prevents NS3 and S4B. These are viral proteins interaction. And as it does that, it prevents the formation of the supermolecular platform that actually does the viral replication. So this is in phase 2. It's a strange study because it's in phase 2 for preventing of disease in contacts. It's almost as a traveler's indication. And Novartis will enter phase 2 soon. So it's a protein that binds to an S4B. So these are the most advanced drugs that will act on this initial phase. So you have to give to patients in the first 48 hours. So dengue has a much shorter viremia than COVID-19. Now, the host-directed therapies are thought to believed to be more useful as the disease progresses, as you try to prevent that shock syndrome, an organ impairment associated with this shock syndrome. And this is usually, as I said, occurs within the 3-4 days. This is when the immune system kicks in, platelet numbers fall as part of the endothelial disease that occurs in dengue. So I will go back a little bit more. So before that, so there are 2 drugs which are actually undergoing clinical trial. That's based on the belief that mast cells are relevant in dengue. So there are 2 compounds, montelukast and leukotriene D4 receptor antagonist, and ketotifen. So there is some evidence that these compounds may be useful in patients. So this is being tried further. The question we'll use here is whether we can use what's called resolution pharmacology to tackle dengue. Going before that, so this is a human and in the microbial world. So obviously, there's a lot of microbes inside us as well. Now, we have to think of infection as one of the possible interactions between us and the microbial world. Actually, it's not the most important one. It's just very irrelevant because it kills us. So that's why we study it. Now, if you think there's about 1 trillion, that's 10 to the 12 different microbes worldwide. We are infected by about 10 to the 3, right? That's 1 to 10 to the 9. So it's not very frequent that we get infected by a pathogen. So our immune system is incredibly good. So anything that actually infects us has found ways of actually, in a way, bypassing our immune system or using it in the benefit of replication. So there's a lot of co-evolution. And when there is not a lot of co-evolution, you have diseases such as COVID, where everything goes wrong, right? So COVID is, lack of microbial human interaction. It's when things are really bad. Now, when we think about microbial interactions, the most frequent is no interaction. We don't interact with most, microbes that are around us, of the 10 to the 12. The most frequent ones we interact with are part of our resident microbiota, being it in the intestine or in the skin, in the lung, the nose, the mouth. And very rarely, you may be infected. More rarely, you get disease and eventually death. So you can think of a pyramid. And it's interesting that we studied human microbial interactions from the point of view of disease, whereas we should be studying it in the point of view of the other way of the most common interactions. So what I say is when we study human social interactions, you don't study it from the point of view of when things go wrong, when people are killing each other. You should study from the point of view of what's more common. So this is immunology actually has a very biased view of human microbial interactions. We study that because it matters, because it impacts quantity and quality of life. So let's see what happens when we interact with a microbe. It may colonize us. It may actually initiate an interaction. It may cause infection. So there is colonization and replication. An infection may cause disease. So this is the first point that's necessary. Infection is different from disease. it doesn't matter to be infected. It matters when you have disease from the point of view of the medical need of things. Obviously, and so this is when vaccines came. So a lot of the vaccines, especially for COVID, they're not very useful in preventing infection, but it's okay if they prevent disease. And so this is something that was believed to be useful from very early on. So what happens when we get infected is that we believe there will be a production of mediators of inflammation. And these mediators, as they are produced, they will actually drive a local containment of infection and drive the immune response that will deal with infection. So this is what we think it's beneficial. Disease occurs when these things are inadequate. Inadequate is a good word because it means nothing. It could mean insufficient, as we see in HIV. It could be excessive and misplaced, as we see in sepsis, where it goes for too much mediators of inflammation in blood, and that's not ideal. Unaltered, meaning different mediators are produced when disease kicks in. So if this hypothesis is possible, is true, it is possible that we actually develop strategies that will enhance immunity or decrease excessive inflammation or altered inflammation, and then have an anti-disease strategy. So we've been trying to study that for the last 20 years. So this is a review in 2010, where you have this concept laid in which you may actually prevent damage from inflammation during infection. And that will not actually change the course of infection itself, but will prevent disease that's caused by infection, right? And this is in the case of arboviral infection. There are lots of studies showing this is a valid tenet. For example, if you block platelet activating factor, you prevent the thrombocytopenia observed in models of dengue. So these are treated. Thrombocytopenia occurs during infection. It's prevented in the absence of PAF or when you give the drug. And if you give the drug, you prevent death. So this gives us a proof of concept idea that it's possible to block certain aspects of inflammation, and that will be very useful to prevent disease during infection. I think COVID, from the point of view of us who work with infection, has been instrumental to prove that concept to humans, right? So basically, now it's clear cut. It is possible to block aspects of inflammation in humans, and this is beneficial to treat infection. So steroids are useful in COVID, kinase inhibitors, anti-IL-6, for example, right? So these are very good blockers of inflammation. It does not change the course of the disease, the course of infection, but it's beneficial to people. Now, with that in mind, can we use the concepts of resolution of inflammation to prevent disease during infection? Can we use resolution pharmacology to treat infection? So what is resolution? This is one of many cartoons. Resolution is about how do you actually clear leukocytes and you decrease tissue damage, or you clear tissue damage once an inflammation occurs. And it has to do with many changes in tissue, including apoptosis of leukocytes, the phagocytosis of these dead cells, which we name efferocytosis, the switching off of inflammation from pro-inflammatory to anti-inflammatory molecules, reverse migration, macrophage reprogramming. These are the crucial steps of resolution of inflammation. Now, what's become clear in the last few years is that this is an absolutely active process. And it's driven by mediators of resolution, or pro-resolving mediators, different names to these molecules. So we are interested in trying to use this resolution of inflammation, which is an active process driven by these mediators, which actually help to return to homeostasis. So resolution is interesting pharmacologically because it is therapeutic by definition. You're not preventing something from happening. You are inducing the treatment. You're inducing the resolution of something. So this is what you see as a medical doctor. People come with disease. So there are many mediators of resolution, from the lipid mediators, which were the first described, but there are proteins, peptide gases, short-chain fatty acids, hydrogen peroxide. This list is increasing. So, but it is an important concept that it's not only about the lipids, it's about a range of mediators, and they do actually do that clearance of tissue and return to homeostasis. So, in the context of chronic inflammation, the idea is that if you have failed resolution, this is borrowed from you, Mauro. You know this, yeah. In the context of chronic inflammation, not only you have excessive number of leukocytes coming in and staying there, but you are unable to clear them. So that failed resolution may lead to disease if chronic. The idea being you can actually try to develop strategies or give the pro-resolving mediators back that will actually induce resolution. Mauro has created this term that I like very much, which is resolution pharmacology. Right, so can we do that to infection? And I will talk mostly about an XNA1 binding to this FPR2, so Christina helped me a lot before. So an XNA1 will bind to FPR2. We can use different peptides based on the XNA1 sequence to mimic its action and induce resolution of inflammation. So is there a role for this pathway in dengue? So there is a lot of evidence that XNA1 is pro-resolving in vivo. This is just some more work. So if you do studies in the absence of an XNA1, if you remove the brake, as we say, you have more inflammation and delayed inflammation. If you give an XNA1 analog, you will make inflammation resolve faster. So an XNA1 does induce resolution in the context of acute and chronic models of inflammation in mice. In humans, for example, in the context of bacterial infection, if you remove the brake, like you do experiments in XNA1 knockout mice, what you see is that these animals inflame too much, and they die of overwhelming inflammation in the absence of an XNA1. On the other hand, if you give an XNA1 peptide, it will actually help macrophages to eat bacteria, kill bacteria, and that's associated with also a decrease in the number of neutrophils. So overall, at least for bacterial infections, you see a beneficial role of giving an XNA1 and a malefic role of actually removing the receptor or the protein. So there is evidence for bacteria. There is evidence in chronic models of inflammation. What about the virus world? So, in models of dengue, what we see is that an XNA1, as you infect the animals, an XNA1 levels drop very rapidly within 48 hours and 72 hours. So what you can do in these animals is do what is called the primary or the antibody-dependent enhancement. So you probably have heard of that. There are 4 serotypes of dengue. Most cases of severe dengue are seen in people who have had dengue before. So the idea being that once you've been exposed to dengue, you have non-specific antibodies that don't prevent infection, but facilitate the uptake of the virus by macrophage, and then you have a greater viremia, and you have greater viral loads, and you have more disease. So you can sort of mimic that in animals, and we see the same situation. So there is a drop in an XNA1 levels, although the mediator is decreased, the receptor is there. So you actually see an increase, an upregulation of the receptor, for example, in the liver, just so you can speak. So the receptor goes up. Now, if you do experiments in an XNA1, so this is a confusing slide, so if you focus on the middle, hematocrit is used as a marker of increased vascular permeability. This is what we see in patients. This is the most important marker of disease severity in patients. As plasma gets out, your platelets get your red blood cells get concentrated, so your hematocrit increases. Now, what you see is that in this model of infection in mice, there is a very rapid and short-lived increase in hematocrit, so there is vascular permeability. You can measure that for example, by Evans blue extravasation. What you see in the absence of annexin A1, so if you remove the brake, you have a disease that is more intense and more prolonged in whatever aspect you have. So there is thrombocytopenia, there is increased hemoconcentration because there is increased vascular permeability, and this is associated with a cytokinemia. So there are a lot of cytokines either derived from macrophages or mast cells. So, and in general, if you remove annexin A1, you see an increase in all of these parameters. Now, the same thing you see when you remove the annexin A1 receptor, which is FPR2. You see an increase in intensity, and it's more prolonged. So disease is always more severe and more prolonged when you remove the ligand or its receptor. So you remove the brake, things go bad. Now, what if we give annexin A1 back? So what we treat animals with is annexin A1 mimetic, which is the peptide 2 to 26 in high amounts. So this is a pharmacological treatment which is started just after infection. It's very short-lived, so you cannot delay it too much. So when you do that, you give the annexin A1 peptide, and again, focusing here, what you see is a very significant decrease in disease presentation, as seen by hematocrit and all of the other parameters. Okay, but annexin A1 does not